Don't watch it with your kids.
You'll have a lot of explaining.
Well, this is how you got here.
Lip's my favorite.
Yes.
I
Yes.
just love him. So.
The first couple scenes, you're like, all right, he looks kind of nerdy. Why is he getting all
Yeah.
these girls? And then he gets to college, I'm like, oh my god.
Yes, please.
Yes. No.
Fancy. Must have a binding machine at work or something.
Yeah.
Tonight class. Ooh.
Yeah, everyone like was training me on how to use it. I'm like, I am not using that well. Like, I actually really like it. So I have to bind like all like the lab notebooks. So actually coming out.
I gave them to you.
They're
Like
they're
2 weeks ago.
testing them do it every year in March. So
Yeah.
this month sucks.
In an academic lab, it's not that strict. EPA shows up every like 10 minutes. Well, I guess we can go ahead and get started. So, as you all probably know, the exams are graded and they are posted. I still have one person from my other class that has to take it, so I'm not giving anyone back yet, okay? I can and probably will give him back on Tuesday, okay, as long as he gets back from Mexico, okay? Donald Trump, I'm not 100% sure. Most of you guys did okay. You know, it's the first exam, so it's usually one of the more tough ones for people, just because you're kind of getting a feel of what I'm expecting, okay? Keep in mind that there are plenty of opportunities for you to do better if you're not happy with your grade. Both practicals people tend to do much better on, okay? Also, everything in lab is designed to really kind of help your grade, okay? And then if all else fails, there's the extra credit at the end of the semester, which does help you significantly if you do it, okay? Not only because it can give you an extra 5% points, but it also shows me that you're really interested in trying. So I will help you along if I need to, okay? I'm not saying you can go from an F to a B, but if you are on the, if you're at a 67, and you do the extra credit, and you come to class, I'll give you a C, okay? So I keep an open mind about these things, so don't, run out of the classroom and withdraw as soon as possible. There's still plenty of time to do better, okay? All right, so with that said, now we're going to go into the most difficult chapter of the semester, microbial genetics.
Oh, what about the better test? What happened?
What?
You said something about the next test is going to be more fluent. This one looks, you said the difficult chapter.
So actually the second exam is the hardest, but people do better on it usually than the first exam because they know what to expect.
Oh.
So I think a lot of people get tripped up by my matching. And you'll get the hang of it. I am a big believer in applied thinking and not just rote memorization. So the whole point of my matching is to get you to connect the dots between things, not just say, oh, here's a word and a definition, because in the real world that doesn't help you. What I recommend, which may come in handy for some of you that don't like the matching, is expand upon some of the answers, right? So a lot of them are 1 or 2 words. If you're not sure how that applies to some of the questions, then maybe write a bunch of bullet points next to that word, and then go back through the list and see if anything else jumps out at you, okay? So expanding upon these things will help a little bit, okay? But you do have to connect the dots a little. All right, so chapter 9, microbial genetics. Genetics is essentially the study of heredity, okay, or how things are transferred from one organism to another, okay? To do that, we need to be able to understand the molecules that are being transferred. And the most important one is something called DNA, okay? So in this chapter, we're going to talk a little bit about DNA, its structure, its function. Then we'll talk about how it can be replicated, and then we'll talk about how a cell can ultimately use it to eventually make protein, which is the main purpose for DNA being there. So, some definitions. So first off, all of the DNA within an organism is what we consider the genome. Okay, so if you hear the word genome, all that means is all of the DNA. Okay, it's just a way of kind of, giving it one word, making it easier to talk about. Now, when we look at bacterial organisms and their genome, they have about 0.4 to 9.4 million nucleotides. Now, you may say, what the hell is a nucleotide? Well, a nucleotide is a molecule, okay, that makes up DNA. DNA is a polymer, which means it's a bunch of molecules stuck together in a chain, okay? And each one of those molecules is a nucleotide. So generally speaking, the more nucleotides present, the more complex the organism can become because it has more DNA, okay? So bacteria, which are relatively simple and basic, we have 0.4 to 9.4 million nucleotides. Compare that to the human genome, which has about 4 billion nucleotides. So you can see we have about 1,000 times more DNA than E. coli. And that makes sense. If you think about a E. coli organism, they're always single-celled and they always look the same, right? You have one E. coli and another, they're pretty much the same for all intents and purposes. Where in a human, you have thousands of different types of cells. Okay? And each one of those cells have different functions and form different tissues and then eventually make a multicellular organism. Interestingly enough, every single cell in our body has the exact same genome. Okay? The only thing that changes are the genes or the pieces of that DNA that are actually being utilized by that cell at any given time. So this is why, if you're complex like us, you need to have a lot more DNA so that you can have diversity. You can pick and choose what you want to use at any given time. Now, humans have about 24,000 genes, and as I mentioned before, genes are just a portion of the DNA that actually codes for something. Usually it's going to code for a protein, but it can code for other things as well, which we'll talk about later. E. coli has about 4,000 genes, okay? So they can make about 4,000 proteins on average. So when you really boil this down, about 85% of E. coli's DNA actually codes for protein, where in humans, only 2% of our genome actually codes for something. 98% of our genome is pretty much garbage. It does nothing, okay? At least not that we understand, at least not at this point in time. So just because E. coli has a smaller genome, it doesn't mean that they don't have the ability to make protein. They are just more concise in how they utilize their DNA. Now, we'll talk about this later on in the semester, but it's important that you understand the difference between genotype and phenotype. Genotype is how we describe an organism and its DNA content. Okay? So the genotype for a human would be these 4 billion nucleotides, okay, or all of their DNA, their genome. But as I said before, that DNA doesn't always get utilized or get used to make protein. Okay? The genes that actually get used to make protein, and then those proteins that are expressed, are the phenotype. Okay? So the genotype is all of the DNA. The phenotype is just the small subset of DNA that's getting utilized to produce something within that cell. Okay? These are often very different things, at least in higher order organisms. Okay? Like I said, every single cell in my body has the exact same genotype. But there are many different types of cells in my body, and they all express a different phenotype. Okay? And that's what allows them to be different. So genetics, like I said, this is the study of inheritance, or how things are transferred, okay? Primarily DNA. So transmission of traits from parent to offspring. But also we like to talk about how these traits are expressed. Again, this brings up that concept of phenotype. What genes are actually being used by that cell at any given time. Then the structure and function of the genetic material, which we'll talk a little bit about. And then how this material changes, or what we often call mutations. How do errors develop in this genetic material, and what does it ultimately mean for the organism? Now, all of these different things can occur both on the organismal level, the chromosomal level, or at the molecular level. Okay? And what that essentially means is, as I mentioned before, multicellular organisms in particular have a lot of different types of cells, right? That's what makes all these different tissue structures and functions and things like that. But as I mentioned before, every single cell has the same DNA, or the same genetic material. Okay? The only thing that changes are the regulations of those genes and what gets turned on and what gets turned off. Okay? And that typically happens at the cellular level. So if you want to make a cell that's found in my skin, so an epidermal cell, you would turn on some genes and turn off some genes. Okay? And that would allow that cell to become a skin cell. If I want to make it into, a heart cell, then I would turn some genes on, turn some genes off until it became a heart cell. And a lot of times this will actually occur deep down in the chromosomal level, and then if you really boil down, you can look at the molecular level. But as I said, at the molecular level, every single cell is the same. It's just at the chromosomal and cell level that things differ, and this is what allows for different cells and different tissues to develop. So genetic material as a whole needs to be able to do two things for it to function properly. Okay? First and foremost, it needs to be able to replicate. Why? Because this is the material that gets transferred from parent cell to daughter cell. So if it cannot replicate properly, it will not be able to transfer itself. Okay? Also, when it is being duplicated, it needs to be done accurately. Okay? If it is not done accurately, then it's going to be useless. Okay? If it doesn't look the same, if it doesn't act the same, then your new daughter cell is going to be all screwed up and it's probably going to die. So it needs to be done in an accurate way. So let's talk about the structure of genetic material. As we've already talked about, the genome. The genome is all of the DNA within an organism. Okay? And within the genome, you can have multiple segments of DNA, and these are called chromosomes. Okay? And this is especially important in eukaryotes, because as we talked about before, eukaryotes have multiple linear segments of DNA inside of them. Each one of those segments is a chromosome. When you look at all of the segments, that is the genome. Okay? For prokaryotes, remember they typically only have one chromosome and it is circular in nature. So the chromosome and the genome in prokaryotes are actually usually the same thing. Okay? Because there's just one of them. Now within each chromosome you will have thousands of genes, and those genes, as I mentioned before, are just segments or pieces of DNA that are used by the cell for something. Okay? Most often they will be what we call structural genes, and these are genes that make protein, okay? And eventually produce the cell. But they can be genes that make RNA machinery, which are important for making things like ribosomes. Or they can be regulatory genes, which act kind of like switches. Turn things on, turn things off whenever the cell needs. Now, the genomes themselves do vary greatly in size depending on the organism. Viruses have the smallest genomes. Okay? It can be up, 4, 5, 8 genes, depending on the virus you're working with. E. coli or bacteria are kind of in the middle. You've got 4,288 genes. And human cells have 20,000 to 25,000 genes, depending on who you ask. So, with that said, there's a lot of DNA. Okay? And as the cell gets bigger, there becomes more and more and more and more DNA. So much DNA that if the cell did nothing and let it just kind of be loose and free, it would be much, much, much larger than the cell itself. Okay? So what the cell does is it usually wraps the DNA around proteins so that it's condensed. Okay? This is the only way that it can actually fit within the cell. So DNA is rarely found completely open. It is usually found wrapped around in coils. Whenever you see that little cartoon diagram with the little X's for the chromosomes, that is condensed DNA. Okay? It's wrapped around many proteins forming that big kind of tight X structure. A little bit more about the chromosomes. As we mentioned before, with eukaryotes, the eukaryotic chromosomes are broken up into linear segments. Okay? They're often branched, that's what gives them that X appearance. They are found within the nucleus, as we've talked about before, and they can be as little as 2 or as many as 300. It really just depends on the organism. Okay? We have 46. Some yeast organisms have 250. Okay? Doesn't make them more advanced than us, just means that they have a lot more chromosomes for whatever reason. They can occur in pairs or singles. They're pairs, this is called a diploid organism. They're singles, this is called a haploid organism. For diploid organisms, they're usually sexual organisms, which means that they get one set of chromosomes from mom and one set of chromosomes from dad. And the cell will randomly just pick and choose what chromosome and what gene it wants to use at any time, and that allows for diversity. Haploid organisms are usually asexual, which means they only have one set of chromosomes. Okay, so they have to just pick the one. There's no other option. And as we said before, they are linear. Bacterial chromosomes, again, are circular. They're usually singular. So a lot of times when we talk about a bacterial chromosome and genome, you can use them interchangeably, okay? Because there's just one. Genes. So genes are a region of DNA that codes for something. As I mentioned before, most of the time this will be useful to make protein, or what we call structural gene. But it can be used to regulate things as well as make ribosomes and other RNA bits and pieces. When you have all of the DNA in the organism, all of the genes, this is known as the genotype. And only the genes that are expressed at that time are considered the phenotype. Okay? Please keep in mind that most of the time, all genes are not expressed at the same time. Okay? In pretty much every organism. The organism picks and chooses what gene it wants at that time, depending on the situation. Okay, so now let's actually talk about DNA itself. Okay, so we talked about how it can be structured within the organism. Now let's look at the actual molecule. So DNA was first described in 1953 by James Watson and Francis Crick. So it's a relatively new molecule. It hasn't been around for that long as far as science is concerned. When it was discovered, it was a really important finding because this is the first time that we were able to understand how things are transferred from one cell to another. Okay? Before that, we really did not know. As I mentioned, they are made up of nucleic acids, and DNA is just a polymer or a chain of molecules. And those individual molecules are nucleic acids. And interestingly enough, DNA is made up of chains or polymers, but those polymers aren't just by themselves. Instead, they're usually stuck to each other, forming a double-stranded molecule. Okay? So DNA is often found in a double-stranded molecule. And once these guys bind in a double-stranded molecule, a slight twist forms, and this then becomes what we call a double helix, or kind of a spring-like shape. So when we actually look at the individual molecules of DNA, or in DNA, these are the nucleotides. And nucleotides are made up of three things: a phosphate, a deoxyribose sugar, and a nitrogenous base. Okay? The only thing that changes from one nucleotide to another is the nitrogenous base. And there are 4 nitrogenous bases that can exist, okay, at least for DNA. Adenine, thymine, cytosine, and guanine. A, T, C, and G. So they're your 4 choices. And that's the only thing that changes in each one of those, and that is the nitrogenous base. When we look at DNA, it's a molecule. You typically write it out like this, where each one of these letters represents an individual nucleotide, more specifically an individual nitrogenous base. And there are special molecule modifications at either end of the DNA. And so at the start of the DNA, we often call this the 5' end, which means there's a 5' modification on the phosphate. And then at the end of the molecule, we often call this the 3' end. Okay? For our purposes, this is important because it tells you the start and the end. Okay? It's kind of like reading a book or a sentence. But as I said, DNA is found in a double-stranded molecule, so you will have nucleotides that can bind, okay, to this other strand, creating the double-stranded molecule. Okay? And interestingly enough, on this bottom strand, it runs in the opposite direction. So this becomes the 3' end, and this becomes the 5' end. So if we were to actually look at this in a molecular way, as you can see here, you can see the individual nitrogenous bases that are different colors. Each one of those colors represents a T, or C, or a G. Okay? As I said, those are the only things that change throughout these nucleotides. Next to the nitrogenous base, you see the deoxyribose sugar and the phosphate. Okay? That stays the same in every single nucleotide. Okay? It does not change. As I said before, it's found as a double-stranded molecule, which you can see here. And what you note is that the nitrogenous bases actually are the bit that stick to each other, okay, to form this double-stranded molecule. Now, these nitrogenous bases only stick to each other in a certain way. So A's always stick to T's, and C's always stick to G's. Okay? So when you're looking here, you see a G, so that means there's going to be a C stuck to it. Here we have a T, that means an A is stuck to it. Okay. Now, between these nucleotides in a single string, so ignore this part, just look at the single string, you can see that they are held together by these lines. These lines are what we call pi bonds. These are very strong covalent bonds. Okay, they're difficult to break. Now, when the two strings or two strands come together, and these nitrogenous bases can bind to each other, they are held together by dotted lines. These dotted lines are what we call hydrogen bonds. And hydrogen bonds are very weak bonds. What this means is that the cell can easily break the two strings apart, but it can't pull the string and break it apart, at least not easily. Okay? And this is important because the string, okay, or the sequence of these nucleotides is what determines its function. Okay? So the way that these are set up in the string, in the order that they are, is what determines what that DNA will do for the cell. Yeah?
Isn't there a U?
So U's are coming up, that's an RNA. Okay, so right now we're just talking about DNA. And RNA, U's will replace the T's. So these nitrogenous bases can be purines or pyrimidines. And as I said, adenine, or A, always pairs with thymine, or T. Guanine, or G, always pairs with C, or cytosine, or C. Okay? And as I mentioned before, these are held together by those weak dotted bonds, or hydrogen bonds, which can easily be ripped apart by the cell. Why is DNA a double-stranded molecule? Well, it's double-stranded because it's much better protected that way. Okay? It's safer for the molecule to be double-stranded. So it's often double-stranded and only is single-stranded when the cell needs to use it so that it doesn't get degraded or break apart. As we've already mentioned, DNA runs in an anti-parallel arrangement. So the top strand, or which we often call the sense strand, goes from 5' to 3'. And the bottom strand goes in the opposite direction, so from 3' to 5'. Interestingly enough, when the cell is using DNA, it only needs a single strand. It doesn't need both necessarily. So you can actually have one gene on the top strand and another gene on the second strand. Okay? This is what allows DNA to be very, very concise. Okay, so that's DNA. Now let's talk about how the cell can actually replicate the DNA, or copy it. This is very important for the cell because this is what allows the cell to divide. When a cell divides, it has one chromosome in the beginning, if it's a bacteria. And then once it decides it wants to go through division, it makes a copy of that chromosome. Why? Because the new cell it's going to produce needs a chromosome. So it makes a copy of its own and then puts that into the new daughter cell. So being able to replicate is extremely important for division. Okay? But please keep in mind that most of the time that the DNA is not being replicated. Okay? It's only when the cell is divided. For division to occur, it requires a whole bunch of enzymes, a whole bunch of anabolic pathways, none of which we're going to talk about in great detail. Okay? Pathways it is. And we're going to whittle down the 30 enzymes to 5. The 5 most important ones. Helicase, primase, DNA polymerase 3, DNA polymerase 1, and ligase. Helicase, which is the first enzyme to get involved, helicase is responsible for unzipping the DNA. What that basically means is it takes the double-stranded DNA and turns it into 2 single strands. As I mentioned before, DNA is not useful as a double-stranded molecule, so it needs to be single-stranded for the cells to utilize it. After you have your unzipping and you have your 2 strings or strands, the next enzyme that comes around is called primase, and this adds an RNA primer to the DNA. What this is just a small little piece of RNA which will then bind to the DNA, and it tells the next enzyme where to start. Okay, so it gives the next enzyme a place to begin. The next enzyme is DNA polymerase 3. DNA polymerase 3 binds to the RNA primer, and it's then going to move down the single-stranded DNA, making it double-stranded. Okay? How does it make it double-stranded? Well, it picks up free nucleotides that are floating around in the cytoplasm, and it adds it to the DNA, single-stranded DNA, until it becomes double-stranded. Next, DNA polymerase 1 comes by, and he removes the RNA that was added before by primase and replaces it with DNA. This is important because you don't want RNA in your DNA, right? You want it to be all DNA. Then lastly, the last enzyme called ligase comes in, and he fills in any gaps that may have been left over, okay? Any kind of holes that may have been left. Once ligase is done, you have a full double-stranded molecule that is strong and can then be utilized by a new daughter cell. So this entire process of duplicating DNA in a cell is what we call semi-conservative replication. All that essentially means is that you start off with one double-stranded molecule, you finally end up with 2 double-stranded molecules, but those 2 double-stranded molecules are half old and half new. Okay? So you can see here the old original DNA is blue, and by the end of the day, we get 2 new double-stranded molecules. Half of it is blue, which means it originally came from the original sequence, and then the other half is pink, which is new. This took forever for us to figure this out. It wasn't until the 70s that we were able to figure this out. Before this, we really didn't know how DNA was replicated. Okay? So this was an important step because it shows us that the DNA is not completely new in a daughter cell. Half old, half new. Why is this important? Well, when we talk about mutations, this is how mutations can actually develop. Okay? Because that old sequence is there, and if the mutation is there in the old sequence, it will go into the new daughter cell as well. So how does this all get started? Well, first off, there needs to be something called the origin of replication within the DNA. And this is an area that has a lot of A's and T's in the DNA. And you may ask, why are there A's and T's? Well, if we go back to our little cartoon diagram of DNA, you'll note that between G's and C's, there are 3 dotted lines. And between A's and T's, there are only 2. What that means our A's and T's are held together much weaker. So if there's a lot of A's and T's, it's much easier for helicase to whittle in and push them apart because they are weaker bonds. So, origin of replication, this is the site of the start of DNA replication. And as I said, it is an area that has a lot of A's and T's. Okay? And that's really the only thing that denotes that. Once helicase finds this origin of replication, It wiggles its way in, untwists the DNA, and then separates those two strands by splitting up those hydrogen bonds. This allows for primers to be added, and then the next enzyme called DNA polymerase III to come in and do most of the work. DNA polymerase III is the primary enzyme that copies the DNA. This is the guy that once this single-stranded molecule is developed by helicase, he will come in, like so, and he will grab free nucleotides and add them to their corresponding nucleotide in the original strand. Okay? And he will do this all the way down the DNA until it becomes double-stranded. It's important to note, though, that he can only go from 5' to 3'. Okay? He can only go in that direction. He can't go backwards. This will become important in a little bit. Now, as he moves down the DNA, this process is called elongation. Basically making a long string of DNA. That's the name. Once he is done, DNA polymerase I comes in and removes any RNA that's there and replaces that with DNA. And then ligase comes in and replaces any, or repairs, any breaks in the DNA that may have formed throughout this process. So if we were to look at this in an actual bacterial kind of example, what we see here is the bacterial plasma, or chromosome, big circular chromosome. And what's happening up at the first example is you have helicase splitting apart those two strings of DNA. Okay? And this creates two double-stranded molecules. RNA primers, which are these little black bars, are added by primase. And this tells DNA polymerase III where to bind and start replication. Okay? The cool thing about bacteria is that they have a circular chromosome, which means they can go in both directions for their replication. This is one reason why prokaryotes can divide much more rapidly than eukaryotes. Because we have to go just in two directions. They can go in four. Now, as I said, DNA polymerase III can only go from 5' to 3'. Okay, so up here it's going to go that way. Down here it will go this way. So that leaves two areas where the enzyme has to go backwards. This isn't possible. So what happens instead is the enzyme will go in short little bursts. Okay, and these create little fragments called Okazaki fragments, as you can see here. These Okazaki fragments are kind of problematic because this is where a lot of errors can develop. But DNA polymerase I is responsible for coming in and making sure that this is done properly and removing any RNA that may be left over. Now, this occurs throughout the entire chromosome, okay? And as it's going, it looks something like this, where you actually have one chromosome here and another chromosome here, okay? And this can go all the way down till the very end. Once it gets down to the very end, you run into a problem where the two chromosomes remain stuck together, okay? Because the enzymes literally just run into each other. So what the organism will do is it will have a separate enzyme called a nickase, which will come in here and it will nick or cut the DNA at both places, and this creates two new chromosomes. One will stay with the organism, the other one will go with a new daughter organism. All right, so that is the basis of DNA replication. I would never expect you to, regurgitate every step of DNA replication. What I do expect you to know are those 5 enzymes we talked about and a working definition of what those enzymes do. Okay? And I expect you to understand that DNA replication is important for cell division. And it is only done during cell division. So small percentage of the cell's life. Only when it is dividing is DNA replication occurring. So you may ask then, what is the DNA there for the rest of the time? Well, the DNA is there for the rest of the time to actually code for protein production. Okay? And that brings us to the next step, and this is what we call the central dogma of biology. Essential dogma of biology basically states that DNA acts as the master template for proteins. And the way that it acts as a template is it gets transcribed into RNA. So DNA gets copied essentially into RNA. And then that RNA gets translated into protein. Okay, so there's 2 steps involved: transcription and translation. This is important because this is how all proteins get produced in the cell. Okay? Without transcription and translation, there's no protein, and without protein, the cell dies very quickly. So let's talk a little bit more detail how transcription and translation actually works. So this is a nice little cartoon diagram. Up here we have DNA. This is the master copy, codes for all of the messages or protein within the cell. This gets transcribed into RNA, and this RNA can come in many different flavors. tRNA, mRNA, rRNA, double-stranded RNA, single-stranded RNA. There's lots of different types of RNA. The one RNA that most people think of, though, when they talk about it, is something called mRNA. And mRNA stands for message RNA, and this is used to actually act as the message for the next step, which is translation. And this is where RNA is converted into amino acids, and amino acids then eventually form a protein. Okay? So the name of the game for DNA, for 95% of the cell's life when it's not dividing, is protein production. Okay? It acts as the template for RNA, and then RNA acts as a template for protein. Okay? So how exactly does DNA get turned into protein? Well, it happens based on something called the triplet code. And the triplet code basically states that for every 3 consecutive nucleotides in DNA, so every 3 consecutive nucleotides, 1, 2, 3, 1, 2, 3, you can get 1 amino acid. 1 amino acid. So 3 nucleotides give you 1 single amino acid. How many amino acids are there? There are 20. Okay? So you can see why we need to have 3 nucleotides to allow for differences and diversity to get up to that 20 amino acid number. So here is an example of DNA at the top, the pink guys. And you can see 3 consecutive nucleotides equals 1 triplet. Okay? That triplet is converted or transcribed into RNA. Once it becomes RNA, it is now called a codon. It's the same 3 consecutive nucleotides. And those 3 consecutive nucleotides eventually code for a single amino acid. So if I have a piece of DNA that has 300 codons, how many amino acids would you expect from that? 100, right? Because you need 3 nucleotides to get 1 amino acid. So, before we talk about transcription, let's talk a little bit more about RNA. Okay? RNA is a molecule very similar to DNA. The major difference, though, is that instead of having a deoxyribose sugar, it has a ribose sugar. Okay? And that's what the D and the R stand for in DNA and RNA. Also, RNA tends to be single-stranded. Okay? It doesn't always form double-stranded like DNA does. And instead of using thymine or T, it uses a U or a uracil. Okay. So if I were to draw a sequence and it looked something like this, you should be able to tell right away that that is RNA because there are U's. Okay? If there were T's, then it's DNA. Also, RNA is not very stable. This is why it is single-stranded. So what that means is that it's constantly being degraded by the cell. Okay? It's not like DNA that's there forever for the rest of that cell's life. RNA can be produced and then it can be destroyed relatively quickly. So one way of thinking about it is DNA is like the library, right? Where you have books and those books are there forever, right? And if you want to take those books, let's say it's a library that doesn't let you actually take the books out, let's say the Library of Congress, right? They don't let you do that. So what you do is you take the book and you Xerox it, okay? The Xerox copy is the RNA, okay? Now, as I said, there's many different flavors of RNA. The one that most of us think about is mRNA, or messenger RNA. This is RNA that codes for the actual protein. So earlier we talked about those 3 consecutive nucleotides equaling 1 amino acid. That is what you would find in mRNA. A bunch of those 3 consecutive nucleotides over and over and over again. So this is the one that actually allows for the protein message to form. tRNA, or transfer RNA, does exactly what its name implies. It takes RNA and transfers it or translates it into amino acids. So you may ask, how do those 3 consecutive nucleotides become an amino acid? Well, that happens using a tRNA. If you look at a tRNA molecule, it is in the shape of a T, which is easy to remember. At the bottom of the T, there's what we call a hairpin, or a loop, and this has something called an anticodon. At the top of the tRNA, we have a tail, and this tail is where the amino acid would bind, okay? So let's say that I have a tRNA for methionine. Methionine is an amino acid. That tRNA would have a methionine stuck at the tail, at the top. And then at the bottom, the hairpin, it would have an anticodon that says UAC. Okay? So 3 nucleotides called U, A, and C. Now, this tRNA has UAC. These are nucleotides that can stick to a corresponding or complement nucleotide. So just like DNA, A's stick to U's, and C stick to G's, okay? So wherever in the mRNA molecule you have an anticodon, or a codon, that binds to this anticodon that has a U, and a G, this guy will stick and bring with it the methionine, okay? So in this molecule of mRNA up at the top, we have our first codon, which has an A, a U, and a G. A binds to U binds to A, and G binds to C. Once this sticks, it brings with it the methionine, and this allows for that methionine to be put into the protein. Okay? So it translates the message by allowing for the tRNA to bind to the mRNA, and it brings with it an amino acid, which eventually can be used in that protein production. Yes?
Okay, how would you differentiate between the codon and the amino acid?
You mean just by looking at it? Or?
No, I mean because you said that the triplet is equal to a codon, but you also said the triplet is equal to an amino acid.
So one triplet gives you an eventual amino acid, right? But the codon is just the region in the RNA, okay, that has those 3 consecutive nucleotides that allows for the amino acid to be come down, okay? So it's not the same thing. An amino acid and a codon, or an amino acid and a triplet aren't the same thing. It's just that is the message that allows for the amino acid to be utilized, okay? Now, how did I know this was a methionine? Well, that's because all eukaryotic RNA starts with a methionine codon, okay? Just the way that it works. I don't have these memorized. Most people don't, okay? I just happen to know this one because every message starts with it. So, another type of RNA is known as rRNA, or ribosomal RNA. rRNA, or ribosomal RNA, is primarily used to make a ribosome. This is important to understand because, as we mentioned before in chapter 5, ribosomes are used to make protein, right? And so if ribosomes were actually made out of protein, who would make them? No one, okay? So most of the ribosome is made out of RNA instead, okay? When we look at the ribosome, it's made out of 2 parts, a large subunit, which is the big guy, and a small subunit, which is the small guy, okay? These two subunits will separate out, and this is what allows the mRNA to bind, okay, in between. Once the mRNA binds in between, you can see that those codons are then exposed, and this allows the anticodon from the tRNA to bind to those regions. So all the ribosome is doing is allowing for this interaction to occur. And you can see that this tRNA, right, brings with it that methionine amino acid at the tail, like I mentioned before. And this starts the process of translation or protein production. So where does RNA come from? Well, RNA comes from one particular enzyme, and that is an enzyme called RNA polymerase II. Okay? Unlike DNA replication, it's relatively simple. You have one enzyme, RNA polymerase II, which comes in, separates the DNA so that it's two single strings, and then it literally goes down the string, making a copy of the DNA in RNA. Okay? One enzyme does most of the work. How does it know where to bind? Well, it binds at a place called the promoter. This is a region in a gene that says, hey, come here, this is where I want you to start. Once the RNA is produced, then the RNA can be used for translation. Okay? Translation is where you convert RNA into amino acids, and those amino acids eventually become a protein. Okay? A protein is really just a long string of amino acids that are folded around itself multiple times. For translation to occur, there's 5 stages: initiation, elongation, termination, protein folding, and protein processing. Initiation is where the ribosome binds to the mRNA. Elongation is where you have that amino acid chain being produced. Termination is where the ribosome leaves and poops out that amino acid chain. Protein folding is where that long string of amino acids gets folded until it becomes a 3D structure, or 3D protein. And then protein processing is where the cell will clip and cleave and make it look as beautiful as possible, so then it can function as a protein. So, to get a protein, there's a lot more involved than just the ribosome. Okay? The ribosome does the first 3 steps, but the most important steps, protein folding and protein processing, are done outside of the ribosome. So initiation, this is where the ribosome binds to the mRNA. In eukaryotes, this always starts with a UAG. This is what we call the start codon, and this always codes for methionine, as I mentioned before. In prokaryotes, it can vary depending on the organism and or the protein. Okay? It's not always the same. Now, when we look at the actual message for an amino acid, or what we call the codon, those 3 consecutive nucleotides, one thing that's pretty interesting is that there is redundancy in the code. And what that essentially means is that for each amino acid, there are multiple codons available. Okay? So if we look here, we have a codon chart. This lists all of the amino acids, okay, and the possible codons that can be used to produce those amino acids. So let's look at leucine, for example. Here we have leucine. If you have a CUU, a CUC, a CUA, or a CUG, any of those combinations will give you a leucine amino acid. And you may say, why the hell is there this redundancy? Why make it more complicated than it needs to be? Well, the reason there's redundancy is if there is some sort of error that happens in the DNA, it may not cause that big of a problem. So let's say I start off with a CUU, and during replication something gets screwed up, and I end up having a CUC. Do you think anything would happen to that organism? No, because they're going to get the exact same amino acid. But if I had, let's say, CUU, and I changed my C to a G, do you think anything would happen to the organism? Maybe, right? Because if I change that C to a G, it becomes a valent. Now, for that protein, that may be a huge deal, or it may do absolutely nothing depending on where it is. Okay? But by having this redundancy built into the code, it makes these types of changes in the DNA less lethal. Okay? And this allows the organism to survive longer. So as we mentioned before, 3 consecutive nucleotides. This makes a codon in the RNA, and this corresponds to the anticodon in the tRNA, and that tRNA then brings down with it an amino acid. Now, as we talked about before, the ribosome is important for initiation, elongation, and then termination. Okay? And this is how the amino acid chain for that protein is produced. I'm not going to go into the details of how translation occurs because it's really boring. But like I said before, all that really happens is the mRNA binds between the large and the small subunit. This exposes those codons, and this allows the tRNA to come in and bind to those codons. Okay? With the tRNA comes amino acids, in this case a methionine and a leucine. Once these come into close proximity with each other, a reaction occurs that allows them to stick to each other, or form a peptide bond. Once this happens, you now have a string of 2 amino acids, and you have a tRNA that is naked. That tRNA leaves, this ratchets down the mRNA, exposing another codon, and now another tRNA can come and bind, bringing with it another amino acid. This can then form a bond with the original string that we produced earlier, and then this creates 3 amino acids. Okay? This happens over and over and over and over again, the whole way down the mRNA, until you reach something called a stop codon. Once the ribosome reaches a stop codon, it stops, opens up, and releases the peptides, okay, or the protein. So what terminates protein synthesis? Stop codons. These are UAA, UAG, or UGA. Okay? Any one of those will work just as well as the other. Now, as I said, translation isn't the only thing as far as protein production is concerned. There's a lot of stuff that happens after translation, and we call these post-translational modifications, or PTMs. Okay? During this, the protein is folded into its structure, and then things are added or cleaved off as the cell sees fit. And this ultimately determines what the protein is going to do. Okay? So these PTMs are just as important as the actual protein production in translation. So here's an example of all of this at work in a prokaryotic organism. Okay? So up at the top we have our DNA, right? This is the purple and pink guy. And we have RNA polymerase, which has opened up that double-stranded DNA and is making a copy of it in RNA, also known as transcription. Now, because prokaryotes don't have a nucleus, everything is happening in the cytoplasm. So almost instantaneously, or sequentially, after the RNA is produced, ribosomes can bind and begin the translation process. Okay? So it binds, it runs down the message, and it creates a peptide chain, which is the beginning of the protein. Okay? Now, interestingly enough, again, because there's no nucleus, transcription and translation can occur pretty much at the same spot in prokaryotes, which means they can make protein very, very fast. And not only can it occur at the same spot, multiple ribosomes can bind to one mRNA molecule in prokaryotes, which means now you can make a lot of protein from one RNA molecule at the same time. Because each one of these ribosomes is going to make one individual protein. So we have got 8 ribosomes, which means I get 8 proteins basically at once. So to make a long story short, prokaryotes are very, very, very efficient at making protein because of these 2 phenomena. Having transcription and translation in the same spot, and ribosomes can bind multiple times to the same RNA molecule. This is another reason why prokaryotes divide so rapidly. Because to divide, you need protein. Okay? And so you have to make that fast if you want to divide quickly. Eukaryotes, on the other hand, are much, much, much slower at making protein. And there's a couple reasons for this. First of all, they have a nucleus, okay? And the nucleus protects the DNA, and so it only allows RNA or transcription to occur within the nucleus. Translation has to occur outside of the nucleus. So for a protein to be made, it needs to be transcribed into RNA in the nucleus. Then that RNA has to leave the nucleus, go into the cytoplasm, and then bind to a ribosome and then start translation. So obviously that's going to take a little bit longer. But another problem that eukaryotes run into is that they have a lot of garbage DNA. Remember in the first slide I talked about how we only use 2% of our DNA, 98% of it is garbage? Well, that 98% of garbage is littered throughout our DNA all over the place. So if we were to look at a gene, okay, it would look something like this. These big bars would be what we call intronic DNA, and this is the garbage, the stuff that is useless, okay, for protein production. The little bars, this is what we call the exonic DNA, and this is the stuff that is useful, okay, in producing the protein. Now, as you can see, the intronic DNA is littered throughout the extronic DNA. When this is transcribed into RNA, it is transcribed exactly how you see it. So you transcribe the good with the bad into the RNA. Now, if this were to leave the nucleus as is and try to be translated, it would not make a protein. Because all this I's, all these intronic crap, is going to screw that up. So the cell has to do one additional thing. Not only does it have to make the RNA and then let it leave the nucleus, it actually has to cut out, or what we call splice out, this crap. So that you end up with this. Now this can actually be utilized by the ribosome to make protein. So you can see why it takes eukaryotes much longer to make protein, because it has to deal with these two problems. It has to let the RNA leave the nucleus, because transcription and translation can't occur at the same spot. And as it's leaving the nucleus, it has to get rid of the introns, or this region of DNA that doesn't code for anything, and only have the exons left. So in a nice little cartoon diagram, this is what it looks like. DNA, we have exons and introns, so good stuff, bad stuff. When we make our RNA, we have both the good and the bad. And as it leaves the nucleus, there are little enzymes called spliceosomes, which cut out the intronic DNA, or the garbage. Okay? If this doesn't happen, the cell will not produce the protein, and it will ultimately die. Now, you may ask, why go through all this extra trouble? Well, it actually does give some benefits to the eukaryote. Sometimes this splicing, or where you cut out the bad stuff, isn't perfect all the time. And so what that means is you can get different splices that occur, and we often call these splice variants. And that ultimately means that you can make many different flavors of the same protein based on this splicing. And that may sound like a bad thing, but in reality, it increases diversity and makes the cell stronger in the long run. Okay? So having this splicing mechanism is important for cellular function in eukaryotes. All right, so how are these genes turned on and turned off? Okay? When we talk about a gene in particular, it can either be an inducible gene or a repressible gene. Inducible genes are genes that need to be turned on. So they're usually in an off position, and they need to be turned on whenever the cell needs whatever they code for. A repressible gene is the opposite. This is where they are always turned on, and they're only turned off when the cell no longer needs them. Okay? Usually when conditions get bad. Now you may ask, how do they turn genes on and turn genes off? Well, in prokaryotes, they do that by having something called an operon. And an operon is basically a region of DNA in the beginning, and it acts kind of like a switch. And the way it does that is a big-ass thing protein, sugar, or something, binds to the operon, and it literally blocks transcription.